Hello, my name is Anand, and this is a 5-minute video walkthrough of our work on spatial transcriptomics of macrophages in lymphoid tissue and DLBCL. I work in the National University of Singapore and I'm a medical oncologist. Our interest is in DLBCL, the most common subtype of non-Hodgkin lymphoma, where macrophages are known to be important, but we don't know the precise molecularly defined subtypes that are possibly prognostically relevant in the setting of chemoimmunotherapy, because most prior work has been done with one or two immunohistochemical markers. So we use digital spatial profiling, the spatial transcriptomic technique from NanoString, which employs UV photocleavable probes to be able to identify region or cell type specific transcripts in DLBCL formalin-fixed samples. We selected for macrophages from lymphoid tissue as well as from DLBCL using a CD68 counter stain. And this was done in distinct spatial regions in the lymphoid tissue. We were able to show that the cell type, cell-specific transcript collection by the DSP equipment was robust, because when we compare our findings to that of single-cell RNA-seq data sets, the genes picked up from CD68 regions did map to macrophages in these UMAP plots, while those picked up from CD20 regions mapped to B cells, and those picked up from CD3 regions mapped to T cells. We did see some interesting differences between the macrophages in the interfollicular regions of the germinal center and within the germinal center itself. The interfollicular regions of the lymphoid tissue and the germinal center itself. This distinction has not been reported before, and further functional work will be needed to understand what it is. We also saw some distinctions between macrophages residing in the dark zone and the light zone of the germinal center itself. To try to understand what these distinctions meant, we projected our findings onto publicly available and previously reported maps of the macrophage subsets that have been identified by single-cell approaches. And with this, we see that the dark zone macrophages appear to be that of a HES1 subtype macrophage. And we focus on these dark zone macrophages because we find them to be the signature that defines these dark zone macrophages to be prognostically relevant in multiple large data sets of DLBCL at the bulk transcriptomic level. And this holds true across many different numbers of genes to define the gene set. This is indeed robust across close to 13 different data sets that we have tested. The profiles of macrophages from patients who go on to have a relapse is also significantly different from those who never relapsed, and particularly in certain hallmark gene sets that you see here. And this particular signature is also prognostically relevant in bulk RNA-seq data sets. But these two prognostically relevant macrophage signatures are not like each other, suggesting that there's yet more to be discovered about what are these subgroups of macrophages and how they may be impacting prognosis. Nonetheless, there may be some therapeutic implications that arise from the findings so far. For instance, that the relapsed DLBCL-like macrophages tend to express an IL-1 beta-like monocyte feature, suggesting possible roles for gevacozumab. So in conclusion, our DSP protocol appears to be robust, and we'd be happy to chat with anyone else who's had experience in trying and seeing how to apply these in clinical material, also to learn from others who've extended this work into other settings. We see that there's some new biology to be discovered in macrophages within the germinal center, and that there are potentially clinically relevant subgroups of these macrophages that impact on outcome. This work was all done by people in my lab and by collaborators. Min Liu is a very talented MD PhD student who led the work on the DSP profiling in collaboration with Claudio Tripodi, a lymphoma pathologist from the University of Palermo, and several collaborators I've listed over here. I'll be around at the meeting for the next few days, so if anyone wants to talk spatial transcriptomics, spatial methods, lymphoma in general, I'd be happy to catch up. Just email me. Thank you.